So now I invite the next speaker, Dr. Leelavathi, for her presentation.
Very good morning, students. And I stand at the midst of a learned community. I feel very proud to be a part of this conference. And I take this privileged opportunity to thank the organizing committee members. And before getting into my topic, that is CRISPR-Cas and genome therapy. That was my topic. Hope this won't bore you because in this current scenario, in each and every house, you will have patients suffering from diabetic or cancer. For this, instead of going for so many complicated therapies, CRISPR-Cas, that is a new technology which made it ease and it also less the pain in the patients. Let's see what is the technology it speaks about. Meanwhile, let me tell what is CRISPR-Cas and why it is so important. For these 2 days, the second day of our conference, right? The first day, so many speakers, they gave you insight about, so what is the therapy and what you have to do, what is the cost of, that is for the treatment and all the things. Whereas CRISPR-Cas, that is the newly emerged technology, where you need not to go for pain-going therapies and all. So that it is the process of editing your genome. Hope you know the genome. So each single cell of the body, it have the entire genome of your body cells. I think you have the basic knowledge of the genome. Am I right, dear students? You have the basic knowledge. We have 23 pairs of chromosomes. Do you agree with me? Yes? Yes? So each chromosome have its own functions. Do you agree with me or not? Yes? Whereas what is the function of chromosome number 5? Anyone in the crowd? So you might be heard about 5p minus. It's in 5th chromosome, the P arm. I hope you know about the centromere and you have a P arm and a Q arm. The chromosome have the P arm and Q arm. Right, children? Right? Whereas if there is deletion in the P arm, it causes a disease in the children's. Anyone, any guesses? Any guesses? It causes cat-cry syndrome. The child will cry like a mewing of a cat. You imagine how it will be. If a baby, it cries like mewing of the cat, how it will be. So that is called as cat-cry syndrome. I hope you know chromosome number 21. If any defect in this, what it will cause? Anyone? Anyone? It's a trisomy. Trisomy 21? Yeah, very good. It's a Down syndrome. In case 18, chromosome 18 defect, it causes Edwards syndrome. Like that, each chromosome has its function. Hope, my dear children, you might hear about sickle cell. Yesterday also, so many speakers, they have quoted about sickle cell diseases. So how the sickle cell is being caused? How it is caused? What's the reason for sickle cell? What is the therapy for that? The person who is having the sickle cell, that is, you imagine the RBC is a biconvex in the shape. If it is a sickle shape, while it is passing through the blood vessel, it will cause much pain for the patient. It is caused because of that is mutation which happened in chromosome number 1. That's what I said in the beginning. Each chromosome has its own function. So if this mutation occurs in chromosome 1, the gene for the sickle cell is BCL11A. BCL11A, that is the gene for sickle cell. So this gene is present in chromosome 1. So that's what I'm talking. So we are in the era where we have much development in the field of biochemistry and molecular biology. Do you agree with me, children? Because the molecular biology and biochemistry is not the same as our ancestors as we studied. It is not like that. Much of improvement is made in the field or in the platform of biochemistry and molecular biology. That's what I said. Because of this increase in the knowledge, it paved the way for treatment purpose. The sickle cell, I said just before, it is present in chromosome 1, and the gene is for the sickle cell is present in chromosome 1, and find the solution for the other. So next for this, instead of, this is a second example for CRISPR-Cas, and yesterday I hope Bhuwana ma'am was talking about RPE65. Ma'am, do you remember RPE? Yeah. So this RPE, that is the gene that is 65, that is present in chromosome 1. Actually, this RPE mutation occurred means the people will have defect in visual cycle. They won't be able to see the image properly. For this, instead of injecting into all the things, that's the old therapy, we have CRISPR-Cas gene therapy. Now it has been evolved much, and you can edit the gene. The mutation part can be deleted, and the corrected gene can be infused in that tool, and you can inject to the person, and easily the treatment is also available for RPE. This is the successive rate of CRISPR-Cas. The next slide is, I will tell you how this CRISPR-Cas works. What is CRISPR-Cas? I said about Gelsinger, I said about RPE, I said about sickle cell. For all these things are important disease where the community is facing nowadays. For this, only solution is CRISPR-Cas9. Now there will be a doubt within you. What is this CRISPR-Cas? CRISPR-Cas is clustered. CRISPR is clustered regularly interspaced short palindromic repeat. I'll show you the image. So this is the CRISPR-Cas. So this is CRISPR is clustered, short, regularly interspaced. You can see the space that is regularly interspaced. The repeated unit will be of 21 to 40 base pairs. BP is a base pair, and for spacer will be 20 to 58 base pair. Where is found? This CRISPR under the where it is found? Any idea, dear students? Where you can find the CRISPR? What it is? I said the acronym for CRISPR, but where it is found? Where this CRISPR is found? Anyone? Anyone? So the CRISPR is a prokaryotic. It is present in prokaryote, not in a eukaryote, but it is widely used for eukaryotic treatment. Kindly listen, dear children, because as someone said, the speaker said, you are the bio-legends. So you have to know what's happening, the current modern technology. So instead of reading in the printed books and reading the past treatments, come to the newer treatment, what is available to you. So the theme of our conference is from the lab to market. This CRISPR is still in the lab, and you have to bring to the market. That is there in the hands of you. Okay. So here, CRISPR is a clustered, regularly interspaced short palindromic repeat. So that is the CRISPR. The palindromic repeat, anyone, what is a palindromic? Anyone, what is a palindromic? Yeah, very good, my dear, very good. So from where, you can see the TC, GC, GC, CG. So if you read from up to down or down to up, it will be the same. It won't change. That is a palindromic repeat. Okay. So here, so the CRISPR, as I said, it is a prokaryote. It is not found in eukaryote. And Cas, it is endonuclease. The CRISPR and Cas, they will bind together. So this is the immune deficiency, that is immune modulation where the bacteria will act. So if we target any disease like a fever or viral infections, our immune system will get activated, right? Am I right, girls, boys, students, am I right? Am I right? Yeah, whereas in bacteria, so what happens, they have modulated to form this CRISPR. So this CRISPR, it is a very clever technology. So it will eliminate the virus. Always the bacteria has been infected by virus. To eliminate the virus from the bacteria have made this CRISPR-Cas. So that's what we are using for human genome therapy. That was amazing and fantasy technology. The scientists and the doctors were see how it works, and they chosen this technology to infuse in human treatment. Okay, so here that is gRNA1 and gRNA2. Kindly listen, this is the technique how the CRISPR works. So the CRISPR gRNA1 and RNA2. The gRNA1, it will form the sequence, the DNA sequence, which is similar to the virus. So, okay, so here the gRNA, it will go and bind with the virus, and gRNA2, it will bind with the Cas, and together they will form a complex. You can find here that is this gRNA1, RNA2, and Cas9. Together, they will attack the target, that is a mutation site. So it will act as a molecular scissor. It will cut where the mutation has been occurred. Mutation, there will cut the gene, the nucleotide sequence. If it's for cutting to act as a molecular gene, it needs a PAM. PAM is the protospacer motif, protospacer adjacent motif. If there is no protospacer adjacent motif, that is the CRISPR-Cas, it won't be able to function, or it won't be able to act as a scissor, molecular scissors. Because molecular scissor is needed to delete or to cut the mutated site. Mutated site cut panadhan, you will be able to infuse the correct edited gene. And you can make the regulations, and you can delete any diseases or whatever mutated site if you delete. So then the correct gene will get infused, so you can treat the diseases. So if PAM is not there, the Cas, it won't act. PAM is essential for this Cas and this complex to act as a molecular scissor. PAM is a palindromic adjacent motif. It is also very essential. So in this, the last I said, DSB is a double strand break. You can make a double stranded DNA, you can make a cut, and you can delete any mutation like deletion. I said 5p minus. In case 5p minus is a characterized syndrome. That is, in chromosome number 5, the P arm, there will be deletion. So if the deletion is there, the child view will cry like a mewing of a cat. That's what I said in the beginning, right? So to remove that, you can insert a new gene for chromosome number 5, and you can edit. So that's what CRISPR-Cas has done. And any doubt in this? Any doubt? Any doubt? You can ask me now. And finally, so this CRISPR-Cas is an innovative development in medical science and in the field of research and development. These innovations improve the accuracy and effectiveness of gene editing methods, opening the door for creation of individualizing therapies. Not only thus, this tool can be used in the field of agriculture. How it can be used in agriculture? Whereas for treating human disease, this tool has been used. That's what I was talking so far. This can be used to treat in the field of agriculture. How it can be used? The same technique can be used in the field of agriculture. Anyone? It's the same thing, you know very well. The crops will be infected with viral diseases, and the productivity and disease ability can be increased using this CRISPR-Cas pathway. Any doubts in this? Thank you, dear children. Thank you. Thanks a lot.